Okay, friends, welcome back. We now begin the afternoon session, and we have Shaeri Mukherjee speaking this afternoon here. Shaeri had done her master's from Calcutta University. She then moved to do her PhD to Albert Einstein College of Medicine. And thereafter, she went to Yale University for her postdoc. She's at UCSF now, and she's a professor there. She does uses a lot of these pathogens, and so she does a lot of host-pathogen interaction studies. And I should say that I didn't know Shaeri before, but when we were trying to sort of think about this meeting, one of her excellent papers caught my attention, and that was on Legionella. And what she showed there was that the toxin that it makes has a tRNA mimicry, and it's actually glycosylates ribosome and inactivates ribosome. And the inactivation is so powerful that it matches the inactivation done by ricin. And so that was really quite interesting. And I don't know whether you'll talk about that work, but floor is yours.
Thank you so much. Can you hear me at the back? Yeah, thank you. First, I would like to thank the organizers for giving me this opportunity. I'm really honored to be here. And as Umesh mentioned, I'm not an RNA biologist. I work on host pathogen interactions. And I will hopefully convince you that pathogens are a great tool to study cell biology. No matter which process you are working on, most likely there's a pathogen that affects that process, and you can use that pathogen as a tool to understand that process. So when it comes to a big animal encountering a small animal, you often think that the size will matter and that the big animal will overcome the small animal and will kill it. But when it comes to host pathogen interactions, this does not matter. So like just think of the pandemic we recently had. So as humans, we have 20,000 genes that encode 20,000 different proteins. SARS-CoV-2 has just 29 proteins, and it killed 7 million people within 2 years. So the question that arises is, if we have so many more cells, so many more proteins than these small pathogens, how come we lose this? When they enter our body, why can't we just eat them up, chew them up, and get rid of them? How is it that these pathogens actually are able to overpower an organism that is so much more bigger than itself? One thing that we think is very important here to understand is that pathogens will replicate very quickly, so within minutes, right? As human beings, maybe roughly we will replicate once every 30 years, right? So that means that these pathogens have a huge advantage of changing their genomes quickly compared to us, right? So it's not that our immune systems don't work and we don't have cells to counter these pathogens. It's because when we develop an immune response, the pathogen can change the response and change their own protein so that the response is not effective anymore. So this is where they have a huge advantage. The other thing to consider is that some of these pathogens, especially viruses, they are so small, they don't even have a translation machinery. So they have to depend on the host to make its own protein. So what do they do then? They hijack a lot of the host proteins and use it against itself. So we are providing the virus all the necessary reagents it needs to replicate inside our cells. So we thought, actually, this analogy that, let's say I want to go and rob your house, right? I will not spend my time in your kitchen and see what you have in your refrigerator. I'll go to where you keep your safe, where you have your money and jewelry, take that money and run, right? That's what pathogens do. They do not have time to waste in the host cell. So what do? They target really important proteins in the host cell that will have a maximum effect in a minimal time. So the output of the effect is very big, and that will allow these viruses and bacteria and even fungi to replicate inside. And I'm not trying to say that all pathogens win. When you walked from the cafeteria till here, you encountered several organisms, and yet you didn't get sick. The reason because most of these organisms can be handled by our cells. These are the few organisms that become pathogens are successful in evading the innate immunity that our cells have to protect us, right? So this is not a new concept, using pathogens as tools of cell biology. And I've given just a few examples of bacteria and viruses that have been used to discover some really fundamental aspects of cell biology. So botulinum toxin produces a toxin called a protease that's called more famously Botox. Like some of you might have heard, you're too young to know about Botox, but when people get old and their skin wrinkles, they put this toxin in their skin. Why? Because this toxin actually cleaves the snare proteins that mediate fusion in the junctions of the nerves. So because these get wrinkled, the protease cleaves those junctions, and then the skin smoothens out because those nerves have no ending. So when they smile, their muscles don't move because those nerves are gone, right? But actually, the Botox was discovered and worked upon by Thomas Chudoff at Stanford, and it led to the discovery of the snare hypothesis as to how membrane fusion happens. So this was one of the major tools he had used to make that discovery. Now, Listeria monocytogenes is a bacteria that once it enters the host cell, it acquires actin tails, and it can move inside the host very fast using actin polymerization. And people thought, how is this bacteria taking host actin and achieving such fast actin polymerization? Turns out that Listeria secretes a protein in the host cell, and when people saw that protein, sequenced that protein and looked, they found that a similar protein exists in mammalian cells called the WASP complex. And that's how the discovery of the WASP complex came into play, where WASP is needed for actin polymerization. Now, as a postdoc, I discovered a new post-translational modification called phosphocholination. And at that time, it was not known what this modification does. But when we found that this modification was caused by a domain, we looked for that domain across the living kingdom. And we found that not only bacteria, but all metazoans, including us humans, have a single copy of that enzyme. And guess what our protein is called? Huntington interacting protein E. So when people wanted to study Huntington protein, they did a pull down. They found that protein. At that time, no one knew what that protein does. They threw it away. But now using bacteria, we know that this protein is capable of causing a new type of post-translational modification. Recently, people have knocked out this protein in Drosophila and found that Drosophila become blind. So this has nothing to do with Legionella. It's endogenous protein in Drosophila. And because we use Legionella as a tool, we can discover really fundamental cell biology that can be applied elsewhere. And similarly, all these viruses have been used. I'll talk a lot about the Rous sarcoma virus in a second. So because the first part of the talk is meant to be more of an introduction, I wanted to use this time to just give you a feel for the field of host pathogen interactions, what people have done and where we are going. So in the second part of the talk, we will talk about actual experiments, how we interpret the data and where we are going from there. But to understand that significance, you have to maybe think about where the field lies, what are the questions that people are asking, and why are they asking that? Why is it important to know these things? So I cannot do any talk without mentioning David Baltimore, because he used retroviruses, Rous sarcoma virus and murine leukemia virus. Both of these viruses, by the way, they don't cause any disease. So this is another plug for basic science research. Like if you do something basic, it doesn't mean that you will not have a role to play in major disease solving. What he did was he realized that this virus codes a viral RNA, that then gets reverse transcribed to a DNA that gets integrated into the host genome. This was a discovery of reverse transcription. I'm sure a lot of you use RT-PCR even now to look for changes in mRNA level. And this was discovered because of this virus. So I think that this Rous sarcoma virus has given us a lot of fundamental discoveries, starting from this very first discovery. So Peyton Rous, back in 1960, I think he did the work even earlier than that. What he did was he took this Rous sarcoma viruses causes sarcoma in chickens. So what he did was he took out the sarcoma, which is this big tumor, he ground it up, and then he put it through a very fine filter, right? He collected the filter. The idea would be that this would not have an intact mass. This would not have any cells. It would have molecules, right? So he wanted to ask if those molecules are enough. And what he saw is that if you inject that filtrate into new chickens, you start getting sarcoma. So this was the first discovery that showed you can genetically transfer sarcoma from one animal to the other. But the actual mechanism of how this happens, why this happens, was not known until Mike Bishop came into the picture. So here's Mike Bishop over here. He won the Nobel Prize in 1989. He was the chancellor of UCSF. And I was lucky because he was the director of the Hooper Foundation at UCSF. And he recruited 3 of us assistant professors. And he always treated us very fancy dinners. So he was very generous. And one thing that he taught us was that be fearless. Life is short. Don't waste your time. Just try to do important things. You might fail, but it's worth it, right? And here's his philosophy. It's in the page of History of UCSF. He said that he learned quickly that viruses of animal cells were ripe for the study by the tools of molecular biology. So he knew that there was some potential in studying Rous sarcoma virus. How this virus causes cancer might lead to something important. What did he discover? He discovered the first tyrosine kinase. So what happens is that this viral sarcoma protein from this Rous sarcoma virus goes and activates tyrosine kinase in an uncontrollable reaction, and that amplifies growth. And he did the same thing which I was telling you about, which is that you take that and see if that is present in humans, right? At that time, there was no sequencing. So what he did was do this painful isolation, mapping and transcription analysis of this gene in humans, and he discovered the first oncogene. So this is the early 80s, right? At that time, people thought that you get cancer from a virus or from the sun or some other external sources. People didn't realize that we ourselves have some genes that can become oncogenic, that can cause cancer in our body. So this is what led to his Nobel Prize. So what about, I've been talking about viruses up till now. So what about bacterial pathogens? So these are also very fascinating. So when a bacterial pathogen enters the host cell, the host wants to traffic it through the early endosome, late endosome into the lysosome where it's very acidic and tries to degrade the bacteria. The reason that these bacteria have become successful pathogens is because they know how to subvert this fate, right? So some, like I mentioned, Listeria, Shigella, and Rickettsia, they acquire actindales and they come out of this vacuole. They break the vacuole and they end up in the cytosol. Some, like Chlamydia, uses the Golgi. Some, like Legionella and Brucella, uses the ER. Some make their own vacuole, like Salmonella and Mycobacterium. And some, like Coxiella, which causes Q fever, actually ends up in the lysosome, but they survive because they know how to pump out those hydrogen ions out of the lysosome. So see, these are all these remarkable strategies that these pathogens are taking to fight back against the host, right? So the fact that they have a very short time means they are targeting most likely very important proteins. And by using the pathogen and understanding what is it that they're targeting into the host, we may be able to uncover something new. So my favorite pathogen of choice, as Umesh mentioned, is Legionella pneumophila. It loves headlines. It started with a bang, and it continues to do so. So the American Independence Declaration was signed in 1776. So, and that was in Philadelphia. And to commemorate that signing, 200 years later, in 1976, they had a big conference where they invited a lot of ex-military men, and they were all put up in a very fancy hotel. And turns out that after the meeting, many people got sick and several people died. And people thought, why did these people die? There must have been a bioterror event happening. So because they were targeted because of the independence. And turns out that this was not some shabby hotel. This was a really fancy, ritzy hotel where ex-US presidents had stayed before. And the CDC, which is the Center for Disease Control, were heroes in this. And they discovered, like detectives, what is it that caused those deaths? And they found it was this bacterium, Legionella pneumophila. Interestingly, when you grow bacteria, I'm sure a few of you use E. coli, you can grow it in liquid culture as well as on LB plate, right? Turns out when they were growing Legionella in liquid culture, it was growing just fine. But when they would put it on the plate, it would die. So there's something in the agar that is killing Legionella. So what they did was they put charcoal on the plates to make it like the charcoal would kill it out any toxicity from those agar. And Legionella started growing. So they isolated the first pathogenic strains of Legionella. The CDC is under a lot of pressure these days. Let's hope they survive because they are heroes when it comes to disease control in US and in the world. So even to this day, if you look, Legionella continues to infect very important people, like this Playboy Mansion outbreak. My postdoc advisor got very excited because he thought he would get a chance to visit the mansion. Turns out that there was a pool party, and they were having these steam sort of like party, and the steam was contaminated with Legionella, so people were inhaling that, and that's how they got sick. Our very own UCSF was sued because one of the hospital patients in one of the hospitals died from Legionnaire's disease, again, from the same idea that these air conditioners hold water, and if that water is contaminated, you can inhale that and can cause that disease. So the New York City has now signed an anti-Legionnaire's bill, which means that all hotels and hospitals have to now check for Legionella periodically so that you don't repeat this. And even recently, 2 days ago, the New York Times reported that there was another outbreak that killed many people. So even though Legionella is not TB or HIV and does not kill millions of people, it somehow loves headlines and it can make its way to the chart. So what is Legionnaire's disease? So when people get Legionnaire's disease, what they do is the Legionella starts growing in the alveolar macrophages, which is these lung macrophages. And if you look at the symptoms, they should remind you of SARS-CoV-2. Most of it is almost the same. And the reason that Legionella is not the same as SARS-CoV-2 is because of the sources of infection. So one key thing that's missing here is person to person. So if I have Legionella, I'm standing here and giving a talk, you guys will not get it. So that's a good reason why the spread of Legionella will not be as much as a SARS-CoV-2. But if you have these contaminated sources, you can get Legionella. So what is the life cycle of Legionella? So Legionella enters the host cell. It has a secretion system like a needle, and it secretes roughly 300 bacterial proteins into the host cell. This is 10% of the proteome. So it has 3,000 proteins, it secretes 300. And what you can quickly see is that there's mitochondrial recruitment around the vacuole, but slowly Legionella changes this vacuole to look more and more like the ER where you see ribosomes on the surface. At this point, Legionella starts replicating, and then it's ready to kill the cell and move out. So if you make a single point mutation of Legionella that cannot make this secretion system, this needle, then that Legionella will become avirulent. It will end up in the lysosome and it will get degraded, which means that this secretion system is absolutely important for the pathogenicity of Legionella. And these secreted proteins are doing something very important for it to survive. So you had such a good talk before me that I don't need this slide anymore. So I'll jump right into the next one. So the idea is that in the initiation step, this is the rate limiting step of translation. You can see in yellow, you don't have to read the names, but you can see here in yellow and green, all these viruses that target all these proteins that are involved in the initiation. Like it gives you an idea how important viruses think the translation machinery is. And I think that makes sense, right? Viruses do not have ribosomes, so they depend on host ribosomes. So of course they will target the host ribosome for their benefit. But it's interesting that they target not only initiation, but they also target elongation, and they target even the end, which is the release factors, right? So why should we care? Because sometimes the mechanisms of how these viral proteins affect these last steps or these steps of translation teaches us something very important about the proteins that are involved in these, right? So again, why are you talking about viruses when you work with Legionella? So is there any evidence that bacterial pathogens target host protein synthesis? Yes, actually, the first known bacterial toxin to be discovered back in 1888, actually, 1888, not even in the 1900s, is diphtheria toxin. Turns out it ADP ribosylates eukaryotic elongation factor 2. This elongation factor ribosylation stops translation. Legionella, as you can see here, also secretes effectors that block protein synthesis. The mechanism is not known that well. Shiga toxin does this, Listeria does this. So it makes it important for us to know what these pathogens are doing to the translation machinery. So this is the first evidence back in 1991 that Legionella inhibits host protein synthesis. They did a very simple experiment. They infected the cells with different amounts of Legionella, and they saw that the protein synthesis rate decreased as the bacterial load increased. But only in recent times, data from our lab and other labs have started to identify these Legionella proteins that block protein synthesis. So there are these three Legionella enzymes, these Legionella glucosyl transferases that glucosylates EEF1A, and that blocks the amino acyl tRNA recruitment. There is a protein called SID-I, which I'll talk about. And at that time, the mechanism was not known, except that it bound to EEF1A. In our lab, we showed this very interesting kinase from Legionella, LEGK4. So normally, bacteria have histidine kinases. Serine threonine kinases are more likely to be found in eukaryotes. But this bacteria was encoding this serine threonine kinase, so we thought maybe it's mimicking some function. So it turns out that it was phosphorylating a single residue on the chaperone family, HSP70, especially the HSC70 the cytosol. What does HSC70 do? So as the nascent polypeptide chain comes out of the ribosome, it needs to be folded correctly, and this chaperone folds it. So by phosphorylating a residue, it blocks the ATPase activity of the chaperone, and as a result, the chaperone cannot fold. So we gave it like unfolded luciferase. After the phosphorylation, it cannot fold it. So what happens is it just gets stuck to the nascent polypeptide chain on the ribosome, and the ribosome doesn't move anymore. That's how it blocks protein synthesis. So the question that must come to your mind is, if one effector, one protein is able to block protein synthesis, why does Legionella secrete so many? Like, what's with the functional redundancy? And we thought about it long and hard. And one reason we thought is that protein synthesis is an energetically costly step, right? Like for one amino acid, you need 4 to 7 ATP molecules. So when there is stress in the cell, let's say there is a DNA damage, right? You don't want to make proteins out of damaged DNA, so you need to block protein synthesis. But the proteins that will escape that block in protein synthesis are the ones that will play a role in DNA damage response, right? Similarly, if you have unfolded proteins in the ER, you want to block protein synthesis, but the proteins that will escape that block are chaperones that will fold those proteins correctly, or apoptotic genes that will induce cell death in case it's out of control. And you need to make sure that the cells die as opposed to make unfolded proteins and become a cancerous cells, right? So that means that depending on what is the initial source of stress, you can have block in protein synthesis, but the outcome may be completely different based on what is the initial reason why you block protein synthesis. So we thought, what if Legionella was achieving different things through these effectors by not targeting the same process, but using these effectors to target different processes and inducing these different responses. And the end game is what they were after, not so much the block in protein synthesis. So in other words, these Legionella transcription proteins are maybe not all functionally redundant. So how do we go about testing this? So a very talented postdoc in the lab, Advait Subramanian, he designed this very simple assay where he took luciferase mRNA and incubated it with rabbit reticulocyte lysate, which has all the essential components needed to support in vitro translation of luciferase, which you can measure easily. And then what he did was he incubated this individually with all the known proteins of Legionella that are known to block protein synthesis. And the idea was if they were all targeting the same step and they were all very similar, they will block protein synthesis similarly, right? All of them would have the same effect in blocking protein synthesis. But he was stunned when he saw that there was this protein, CDI, that blocked protein synthesis in picomole levels. And in context with what Umesh was saying, this is comparable to ricin. For those of you who do not know what ricin is, it's a very potent toxin that can kill people very fast. In fact, it's estimated that ricin can inactivate 1,500 ribosomes per minute. That's how fast it is. So we thought that this is very cool. This is a very potent toxin. The other toxins are also good, but this one stands out. So what's with this? Like what is this doing in the cell? And we wanted to know what was already known about CDI in the literature. And we came across this paper where they showed that if you put CDI in a galactose inducible vector, when you express it in yeast, you see that in glucose, obviously CDI is not expressed. You see the yeast is growing just fine. But as soon as you add galactose, yeast dies, right? And you see a block in protein synthesis. And they also showed that it bound to this elongation factor EF1A. So, but we thought that this is not enough to explain the potency, right? Picomole level, this protein is acting on. There's so many molecules of EF1A in the cell. Even if it binds to some, it shouldn't be that potent, right? So what is really happening? And so I will end my first talk here with 7 minutes. I'm on time. And in the next talk, I'll focus on now the more relevant data, like what is CDI doing? What is the mechanism, and where do we go from there? Thank you. Yes. Yeah. I'm just excited to know. Yeah, that's a great question. So normally Legionella, and the natural host of Legionella is amoeba. So in fresh water, it stays inside amoeba. It can survive outside also. It's a facultative pathogen. But we see that whatever Legionella secretes in amoeba, it also secretes in human cells, right? And in all human cells. The interesting thing is the reason they call Legionella an accidental pathogen is normally pathogens evolve across the mammalian kingdom, right? So they go from small mammals and then they eventually, but because of this air conditioning problem, it went straight from amoeba to humans. So I consider it an excellent cell biologist because it learned its cell biology from amoeba. But it has not seen the immune system, right? Amoeba doesn't have one. So in macrophage, what happens is if you and I get Legionella, we have a robust immune system, so we will induce pyroptosis, which will kill the bacteria. We will have a mild fever, a Pontiac fever, and we will be treated with antibiotic. It will be fine. The people who die from Legionella are ones who are immunocompromised or older people whose immune systems are not great, and that's when it can quickly, like, build up a lot of mass in the lung, and that's when it kills. But in the sense of whether it realizes which host cell and can secrete different effectors, that's not the case.
Yeah, Leo.
Yeah, 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 yeah.
Okay, now
No, we haven't. And I will tell you why. So they are not secreted all at the same time. So that is why we didn't combine them. So the initial ones, so Legionella goes through this phase. In the initial 1 or 2 hours, it's not replicating. So it wants to establish a base, right? So it's targeting things that are like relevant for its survival, right? Towards the end is when it wants to kill the cell because it has replicated. And that's when it secretes CDI. And I will tell you more on that in the next. Great question.
So you think that SID1 binding to elongation factor 1 is just, they are not necessarily doing anything?
No, it is doing something. I will show you. So you gave away my punchline, but I still have some data that you didn't say in the introduction. Yeah.
So any more questions?
Yes.
Yeah.
No. Yeah, they have several targets, right? Like, so remember I was showing that it rewires the endocytic pathway because it moves away from the lysosome. So there are effectors that stop that fusion of the initial vacuole to the lysosome. There are, Legionella does not have a nucleus, it's a bacterium, right? Guess what? It has histone as methyltransferases, which means that those are specially made for the host, right? So when it comes, it goes to the host nucleus, this histone methyltransferase, and methylates host histones and affects host transcription. So then it has effects on mitochondria, it affects ER mitochondria contact sites, it affects ER morphology, it affects those 300 effectors are like a repertoire of cell. That's why I said, if you are interested in a particular pathway, come talk to me. I'm sure I can find you an effector that affects that pathway. It does have histone methyltransfer. Yeah, 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 yeah. So the people who first discovered this histone methyltransferase just showed it first in amoeba and then moved it to the mammalian system. Yeah.
Question. So how the Legionella knows that it has to only pump in a select set of effector proteins? I mean, so it is going to have both its own protein and the one
Yes,
going to
yes,
go to the.
I understand what you're saying. So how does it know which proteins to secrete and which proteins to keep inside? So it has a secretion signal in its C terminal. All the proteins that are secreted is recognized by the secretion signal. And remember I told you there's a needle. So that needle will recognize that secretion signal, and those are the proteins that will come out into the host cell.
Right, but how it is going to select among the proteins that it has synthesized, something should be secreted into the target. So let's say it is making some 1,000 proteins. Out of the 1,000 proteins, maybe 50 proteins will go as an effector towards the host, right? So how is it basically, shortlisting those 50 proteins that it has to target?
So over a period of like millions of years, it figured out which are the effectors or which are the proteins that it needs to secrete, right? So those are the proteins that only those proteins have the signal sequence. So it has 3,000 proteins, right? only those 300 will have the signal sequence. The rest are not. So the other thing that you might want to know is, how does it know that it's in the host to make those proteins in the first place? Turns out it's always making those proteins. It's always ready, right? So we've tried very hard in like liquid cultures to get it to secrete so that we can like manipulate the secretion system in vitro. But somehow it's making the proteins, but it's not secreting. So there's something that happens in the vacuole, most likely pH changes or other host proteins, that triggers the release of these proteins. It makes it, but it keeps it until it's in the host. Yes.
Yeah. So you have shown in your like that schematic that it recruits the ribosome on top on the vacuole. So if it is secreting some toxin to deactivate the ribosome, why in first place is recruiting the ribosome? And you also talk something about mitochondria. I didn't get it properly.
For the data, I will come to it. Like, so it has reasons why it's recruiting those ribosomes. So I will maybe give you just a little hint of what's happening. So those ribosomes are translating, the ones that are on the vacuole. The ones that are in the cytosol and on the ER are shut down.
So since these pathogens are targeting elongation, but the elongation is quite similar in both the bacteria as well as the eukaryotes. So how does the specificity comes in?
Again, a super good question. Turns out that in the same operon where you have CDI, there is a protein called MESI. And MESI is the antitoxin that binds to CDI and keeps it inactive in the bacterial cell. If you make a knockout of MESI, Legionella doesn't grow because it kills itself, right? So there is also another mechanism which I will talk later. But yes, it is very careful in terms of keeping the toxin inactivated until it's ready for release and acts only in the host cell and not on itself. I can start? Yeah. All right. So what is the mechanism? Now we go into a little bit more data. So I was really worried that we will never find the mechanism, because what is a question that is sometimes very hard to answer? Like, what is it doing is something very hard to answer. But we thought that structural biology might be something that will help us understand. And sometimes the universe has unique ways of telling you something is going to happen. I ordered chicken wings, and it came in the shape of an inverted L shape. So normally chicken wings, for those of you who eat chicken, are like this, right? My chicken wing was like this. And turns out that CDI is also like this, just like my chicken wing, an inverted L shape. And I'm sure that you guys know what this shape reminds you of. It looks like a tRNA. And this was done in collaboration with this really talented postdoc, Lan Wang, who has her own lab in Hong Kong, with Peter Walter, who was at that time at UCSF. And Lan solved the cryo-EM structure of CDI and found these 3 domains that had made this sharp 90-degree angle, which is not commonly found in proteins. This is something that caught our attention. And when we overlaid the structure of CDI to a tRNA, we found that there was remarkable similarity in the way CDI was folding compared to a tRNA molecule. Now, we knew that if we tried to publish this, we would be completely ridiculed and thrown away. So we wanted to make sure we have more data to say that this is a tRNA mimic. So the first thing Lan and Advait did was to look at the height and width of the molecule, and it was very similar to a tRNA. The other thing that was touched upon in the last talk was these release factors, right? So they occupy the A site and helps in the termination process. So we asked if this CDI is similar to these proteins that bind to the ribosome. And turns out that these proteins have this very positively charged domain that makes contact with the ribosome, because the ribosome is negatively charged. And we found that exact same domain in CDI. So this gave us some hope that it is possible that CDI is acting as a tRNA mimic. And this was very nicely explained again, so I don't have to go in the details. But what is important here to note is that EEF1A is the protein that brings tRNA to the ribosome. Remember, I said the first paper on CDI said that CDI binds to EEF1A. So it's that same protein that binds to tRNA is now binding to this protein CDI. So we thought, if this is indeed a tRNA mimic, does it bind to all the proteins that a tRNA normally bind to? So we did a mass spec analysis. So what we did was we purified GST CDI, and we incubated it either with rabbit reticulocyte lysate, which is where we found our original data, or with HEC293T lysate. It's a human cell line, so we wanted to make sure there's nothing specific happening with rabbits. And we did a pull down and mass spec analysis of all the proteins that bound to CDI. And you can see here on top is the hit EF1A, which is the protein that brings tRNA. But we found all these tRNA ligases and these ribosomal proteins. Immediately, we thought these are very sticky proteins, right? That they come down with a lot of mass spec results. So how do we know that these are real? So obviously, we did a control with GST, but we used very stringent pull down conditions, and we did a Western to show that indeed this was coming down with the proteins with GST CDI, but not with GST. So all these tRNA ligases are coming down with these with CDI, the tRNA splicing ligase, EF1A, all of them come down with CDI, but not with GST alone. So this gave us a little bit of breathing room, thinking that we might be on the right path. So, but we thought if these proteins are binding, especially the ribosomal proteins, then they should be binding to the ribosome as a whole, right? Like they should not be binding to individual proteins at a time. So can we actually pull down the ribosome with GST CDI? And that's what Lan did. She used purified monosomes and incubated these monosomes with either GST or GST CDI. And she found these ribosome large subunit, small subunit proteins coming with CDI, but not with GST. And when she put it on the grid, she could see evidence of the monosome coming down, suggesting that CDI could be occupying the A site on the monosome. So this does not explain why CDI would be so potent, right? There are 60 million, approximately 60 million tRNA molecules in the mammalian cell. And Legionella secretes very little amount of protein. So if it were to compete for A site on a ribosome, it has no chance. Like the host has way more tRNAs than this little tiny protein. So we thought something else is happening with CDI. And when we looked at the structure, we saw that actually in this inverted L shape was hidden an enzymatic domain, which was remarkably similar to another protein from Mycobacterium, Corynebacterium, sorry, called PIMB-. And this was characterized as a glycosyl transferase with a B fold. And when we did the overlay, we found that this was remarkably similar. So we think that CDI is an unusual tRNA mimic where it has coupled the shape of a tRNA with the enzymatic activity of a protein, right? So we wanted to confirm that this is true, that it indeed can act as a glucosyl transferase. So the assay we did was to basically use the GDP mannose or any sugar and then add the transferase and see if GDP is released. And when we did it with different sugars, we saw that CDI was not able to cleave UDP glucose, but it was able to cleave GDP from GDP mannose and GDP fucose. So it has some specificity for these sugars, but not for glucose. Whereas this other Legionella glucosyl transferase actually uses UDP glucose, but not mannose and fucose. So they are different in the way they act, right? Though they are all glucosyl transferases. So then the main question remains, what is CDI targeting? If it's an enzyme and if it's a glucosyl transferase, what is it glucosylating? So what we did was we purified GST CDI, or the catalytic dead mutant that cannot mannosylate, and we incubated with GDP mannose. So it mannosylates whatever is the substrate on these ribosome extracts. And we use concavalin A, which is a lectin that binds to mannose. So the idea is if CDI manosylates proteins on the ribosome, then this pull down will bring those proteins down. And we made sure to use this negative control because if they were pre-manosylated, they will come down with both. But if they're manosylated only with CDI, they'll come only with CDI, but not with this catalytic dead mutant. And that's exactly what we see. The wild type brings down these ribosomal proteins that are manosylated, but the mutant does not, suggesting that CDI is targeting the ribosome and many of the components of the translation machinery to manosylate it. And when we did a mass spec to look for all the manosylated proteins, and because the structure of ribosome is solved, we put these proteins and placed them on the structure of the ribosome, and we found that these are proteins that occupy the A site, the P site, and the exit tunnel, which means that it was basically going just like a tRNA, and then as it was going through the ribosome, it was like painting the ribosome with mannose. And why? Because then the other tRNA molecules the host has can no longer access those ribosomes. So it goes from ribosome to ribosome to ribosome. It doesn't need to stay there. It doesn't need to compete with the tRNA because of this enzymatic domain. So the idea was that we also used EF2, which is known to also bind to the ribosome, and we saw that it was by modeling this protein to where EF2 binds, we found that there was a groove that could explain where CDI would bind on the ribosome. So here's the model, right? If CDI was competing against each tRNA by occupying the ribosome, then you would need the same stoichiometry of CDI to tRNA. But if it was the enzymatic model, and this is the enzymatic reaction, which is to manosylate the ribosome and then go from ribosome 1 to 2 to 3 to 4, then you no longer need the same stoichiometry, and then the toxin can become more potent, right? So how do we test these two hypotheses? So the idea is that CDI is a tRNA mimic, but it also has an enzymatic domain. How do we uncouple which is more important, the tRNA mimicry or the enzymatic domain? The problem of truncation is that the enzymatic domain is in the tRNA fold, so you cannot really remove the entire domains, but you can make point mutations, right? So that's what we chose to do. So by looking at the same translation assay that I had shown before, Advait found that the wild type was blocking protein synthesis at the 50% of protein synthesis was blocked at this 7 nanomolar region, whereas the catalytic dead mutant was blocking protein synthesis, but it was 1,000 times less efficient than the wild type, which means that this enzymatic activity is needed to make this enzyme more potent, but the structure or the shape of the tRNA molecule, it also is important, but it will act at a very high concentration as expected. Which, sometimes when you do these experiments, you think carefully that what if this mutant is not completely catalytic dead and that there is a very slow reaction of manosylation, and that is the reason that you're seeing this very high dose response, but not really because it's a tRNA mimic. So how do we distinguish between the two possibilities? So what we did was we just incubated the protein for a longer time. And the idea was that we took, for 50% translation block, you need 7 nanomolar of wild type and you need 7 micromolar of the mutant. And we took these concentrations for both of them. And see, both of them started at 50, but over time, the wild type went down to 0, but this mutant stayed at 50. So if it was a weak enzyme, it was still go down. It will take longer, but it will still go down. The fact that it's not going down is because this inhibition is caused by the shape, not for the enzymatic activity. This is our way of showing that both of those are needed. But we wanted to uncouple the two. And the way we thought we could do that is we could make mutations in the residues that bind to GDP mannose based on the enzymatic activity. And we could make mutations on that positively charged loop, which I said was the one that makes contact with the ribosome. So we are not making major truncation mutations. We are just simply mutating these positive residues, these lysine to alanine. And this mutant will have an intact enzymatic activity, but it will no longer behave like a tRNA mimic because it will not have access to the ribosome. And then we tested whether the mutants were acting like the way they should. Indeed, if you make mutations here, you don't see the GTP release, which means that the enzymatic activity is down, whereas this K-loop mutant has a very good enzymatic activity. And when we looked at ribosome binding, we saw the opposite, which is that this K-loop mutant, which has good enzymatic activity, does not bind to the ribosome, whereas this mutant, which has almost zero enzymatic activity, binds to the ribosome just like wild type. So now we have uncoupled the two. One mutant binds to the ribosome, one mutant has enzymatic activity. And we ask which one of them is more potent in blocking protein synthesis. And the answer is all of them are good in blocking protein synthesis, which means that even though the catalytic mutant is obviously much more efficient in blocking protein synthesis, combine the potency of the tRNA mimicry along with its ability to act as an enzyme on the ribosomes. So the question as cell biologists is, if Legionella causes mannocylation to ribosome, what does it gain from it? Why is it using so much energy to mannocylate ribosomes? What's the reason? So what is the cellular consequence of this mannocylation? And when we looked in the literature, we found that if you have low-dose elongation inhibitors, it causes stalling of the ribosomes on the mRNA. And as a result, the trailing ribosome will go and hit that stalled ribosome and can cause a collision. And that collision can cause a stress response that is known as the ribosome-associated stress response. So we wanted to ask if Legionella actually was after this ribosome-associated stress response and its downstream consequences. So how do we show during infection that Legionella is causing ribosome collisions? So we thought of two ways in which to show. One is that if you see the block in protein synthesis with CDI or the catalytic dead mutant, you can see that CDI has a very strong effect on protein synthesis, but the catalytic dead mutant looks just like GFP. But when ribosome collisions happen, there are ligases that have to ubiquitinate those ribosomes and get it out and clear that mess. And we see evidence of that ubiquitination only with CDI, but not with the catalytic dead mutant, suggesting that ribosome collisions has happened. But for me, the more important experiment was this one. So normally, when you see the polysome profile of ribosomes, you see these polysomes, and you see that CDI has a lot more polysomes because they're obviously stalling. This is the regular polysome profile. But the idea was that if we treat with RNAs, what happens is that, let's say there's a polysome here, a monosome here, a monosome here, a monosome here on the mRNA. If you add RNAs, the RNA between the two will get degraded. So all of these monosomes will come together and form one monosome peak. But if they have collided, these two monosomes will protect that small RNA in between, and that RNAs will not have access to that. And instead of monosomes, you will get a disome peak. And that's exactly what we see with the wild type. That disome peak, it's very hard to get, by the way. This is very transient reaction, but Advait is super, and he could figure out conditions to show clearly that these disome peak exists. And so this is where we're thinking it happens, that once the mannocylated ribosome stall, the ribosome behind it hits it and causes a collision, and these collisions then induce a ribosome-associated stress response. So we wanted to know what that stress response was like. So it is known through work with Rachel Crean, who is going to come in the next week, that you can have these small molecules like anisomycin and didemonin B that can also cause ribosome collisions. So we used that as a control, and we infected cells with either the wild type Legionella or the mutant that cannot secrete any effectors into the host cell, and compared it with anisomycin and didemonin B treated cells. The idea was these cells would induce a ribosome-associated stress response. This one will, but this one won't. So we looked for genes that were common between wild-type anisomycin and didemonin B, but not .A. And we found 75 commonly upregulated genes. And when we looked at these genes, we were very excited to find this particular gene, ATF3, this transcription factor. Why were we excited? Remember I said that when you have unfolded proteins in the ER, there's block in protein synthesis, but some proteins will escape that block. Turns out ATF4, which is very similar to ATF3, is a beta zip transcription factor, escapes that block during unfolded protein response. And we thought, what if by chance, through the use of Legionella, we discovered a similar mechanism where ribosome collision is detected in the cytosol, and this transcription factor is the one that is communicating that signal to the nucleus. And so the idea is that if you are saying that CDI blocks protein synthesis but induces gene expression, it doesn't make sense, right? If you induce gene expression, what good is it if you don't make that protein? So the ATF3 should override that block in protein synthesis. And that's exactly what we tested. You can see here that in wild-type infection, later time points, you start seeing more ATF3, but you don't see that in .A. And if you remove all the translation inhibitors, you don't see that. But as soon as you just add back, just CDI, not those other four, just CDI, you see this robust accumulation of ATF3, translation of ATF3. But if you take these 5 individually and you put them in the cells, in mammalian cells, just transfect them, you see only CDI will induce ATF3, even though these effectors are blocking protein synthesis, which means that ATF3 is not looking for block in protein synthesis, it's looking for ribosome collisions. And that is why only CDI is inducing ATF3. And we wanted to check if this happens during infection. So here in green is Legionella. This is uninfected, so there's no green. And red is ATF3, there's hardly any ATF3. As soon as you infect the cells with Legionella, you see here Legionella, and the cells start accumulating ATF3 in the nucleus. In .A, there is not much. In the delta 5, also not much. As soon as you add back CDI, it again is robust, which means that ATF3 is transcriptionally upregulated, but also protein level is upregulated, and it goes to where it should be as a transcription factor, which is in the nucleus. And remember, I said that I used Legionella more as a tool to understand basic cell biology. So it turns out what we discovered with Legionella holds true for anisomycin and didemonin B, and even this compound donavalin, but not for cyclohexamide and harringtonin, which also blocks protein synthesis. So this is a specific effect you see for effectors or block in protein synthesis that causes collisions and not regular block in protein synthesis. So then the question finally that we wanted to ask is, what signals allow ribosome stalling to accumulate ATF3? Ribosome stalling is happening in the cytosol. ATF3 is getting made. What is the signal in between? And again, we looked in the literature, Rachel Green and others have shown that if the collision is less, then this enzyme, this kinase ZAC alpha recognizes collided ribosome and will activate this pathway where this kinase GCN2, which is a kinase of EIF2 alpha, gets phosphorylated and you block protein synthesis. But if the collision is very high and the cells think there is no way of turning back, then the same ZAC alpha will induce another kinase, MAP kinase, and this will induce P38 and junk, and this will induce cell death. So we wanted to ask if Legionella was causing this pathway or this pathway during infection. And so we tested that with the help of MEFs that do not have the ability to phosphorylate EIF. So this is the amino acid that gets phosphorylated, and we have it mutated to alanine. So this cannot induce an ISR response. And when we have that, we see that it doesn't matter. Anisomycin is still getting made. But if you look for tapsigargin, and then you see that it's not getting made because this is ISR dependent. ATF4, like I said, is made with tapsigargin with the wild type, but not with the catalytic dead, because this depends on the ISR. And again, we used another molecule, which Peter Walter described, called ISRIB, which bypasses the need for EIF2 alpha phosphorylation. And we see that even in the presence of ISRIB, you get a lot of CDI to be phosphorylated. So finally, we wanted to see if ZAC alpha gets phosphorylated, because that's the cell death pathway. We did an unbiased proteomics of cells infected with wild type or .A and looked for evidence of kinase signaling, and we found indeed phospho P38 and junk were activated with wild type, but not with the .A. So we confirmed that with Western blots. So in uninfected cells, you don't have much ATF3, you don't have phospho P38, and you don't have phospho-ZAC. When you infect the cells with wild-type Legionella, you see evidence of phospho-ZAC, robust activation of phospho-ZAC, robust activation of P38, and also ATF3. So we wanted to know which is upstream of what. So we knocked down ZAC, and we saw that when you don't have ZAC, you don't have P38 and you don't have ATF3, which means ZAC is upstream of P38 and ATF3. But when you knock down P38, you see that you still have ZAC, which means obviously ZAC is upstream, so that's why it's not affected, but you see very little ATF3. So it goes from ZAC to P38 to ATF3. And finally, we wanted to ask what is the significance of this for Legionella infection. So when we look at cell death, we see that when you transfect the cells with CDI, even though CDI blocks its own translation in the cells when you transfect, very little CDI is actually made, but the cells die in 72 hours. But if you make an ATF3 knockout cell line, then the same CDI is not growing because there is no translation. Of course, the cells will not grow, but it's like becoming cytostatic as opposed to growing cells with the negative control. So we wanted to ask if this is the case for Legionella infection. And so we knocked down ATF3 in cells that could be infected, and we found that in wild type infection, 50% of the cells die, but in .A, it looks just like uninfected. But when we knock down ATF3, we can see that most cells don't die at this time point. By the way, this is at 8 hours post infection. So the idea is that at that point, it's still protecting. But if you make the cells grow more and more Legionella, obviously at some point it will still die. And we can see that with the help of the mutants, which is that the delta 5 that lacks CDI does not kill, but if you add back CDI, it kills. And we see the same with the ZAC inhibitor and the P38 inhibitor. So in summary, what I showed you is that Legionella is a very clever bacterium. It enters the host cell. It uses a type 4 secretion system to secrete many effectors. One of them is this protein CDI that looks like a tRNA. And because it looks like a tRNA, EEF1A brings it to the ribosome. And when it accesses the ribosome, it manosylates the ribosome with the help of GDP manos. This causes these ribosomes to stall and cause collision. And this collision recruits ZAC alpha to this collided ribosome, activates P38, and then P38 induces ATF3, which then many of the genes that I showed you commonly upregulated with anisomycin and diadenominin B is actually regulated by ATF3. So this is the master regulator. And then we think Legionella uses this to kill the cells, to disseminate and go on to kill other cells. So I want to thank all the people who I think I already thanked, but also the lab and the funding. And last but not the least, I want to thank all the organizers for this really amazing meeting. I'll go with lots of happy memories. So thank you for hosting.
Plenty of time for discussion. Yeah, go on.
Yes. Yes, so that's a good question. So turns out that most of the mannosylation or glucosylation that happens in the cell happens in the Golgi and the ER, which are membrane bound, right? So as a result, the deglucosylation enzymes are also present in those membrane bound organelles. So Legionella figured that if it targets the proteins with phosphorylation, there will most likely be a phosphatase that the host will try to remove. So it targets with a modification that normally is not seen in the cytosol. That's why the reverse modification is not an option that the host has.
Yeah. And a second question. So when we are talking about SID1 being going from one side to the other in the ribosome, so we expect the substrate, which will be the sugar, to be present all the time.
Yeah.
So how would that work out when it is in the ribosome?
Yeah, that's a good question. So I think that when we do the mass spec, we combine all the ribosomes. So it's possible that not the same ribosome has all the modifications. So it's possible that, you know, it's a heterogeneous mixture of different ribosomes having different manners. So maybe not all are happening in the same exact ribosome. Yeah.
Mimic acts as a zip code, right? So when you have the point mutants that disturbs the tRNA mimicry, do you see less localization of the protein to the ribosomes? Where was the zip code idea coming from?
So we think that by pull downs, we saw that ribosomes were not binding once we mutate that positively charged loop. But EF1A, for example, that is still a substrate for, because that's what Umesh was asking. EF1A is also glucosylated, right? So if all of the EIF1A is glucosylated, even if it doesn't access the ribosome, these accessory proteins that get, and then some tRNA ligases as well, then the host tRNA might not have those resources that it needs to access. That's why the point mutant actually is not as bad as the catalytic dead. The catalytic dead mutant is 800-fold reduction, and the point mutant is 100-fold reduction. So there is some sense that the catalytic domain is much more important than the tRNA structure.
Hello. Yeah. So.
Oh yes, I have to show you. I promised you. So the thing that I didn't show is how is ATF3 escaping the translation block, and we took the 5' UTR of ATF3 and put it on M-APPLE, and we see that's enough to actually turn on translation when you add anisomycin. So the information that allows ATF3 to be made is actually in the 5' UTR. And then there are ORFs in the 5' UTR. We mutated that. We saw that had no effect. So it's something else that's in the 5' UTR, which we are now working on to understand what allows ATF3 to be made when everything else is shut down. And then to answer your question on the ribosome. Yes. So puromycin is a marker for active translation, right? And you can see that in steady state, you see puromycin in the cytosol, in the cell. Yeah. In DOTA, you see the same, like you see these puromycin, which means there is no block in protein synthesis. But when you infect the cells with Legionella, you see that at 2 hours, there is no problem. At 4 hours, there's no problem. But at 8 hours, you see that all these ribosomes on the wild type Legionella vacuole recruit puromycin, which means that they are translating. The rest of the cell, look, the rest of the cell is almost dark, but the ribosomes on the surface of the vacuole is somehow protected from the block in protein synthesis.
Hi. Like, so what is the requirement of it to recruit to the vacuole?
Yes, so that's also another good question. We had another paper on it. So Legionella first starts with interacting with the smooth ER, and then it uses some of the host proteins like BAP31 to hop from the smooth ER to the rough ER, and then it induces a ER-FEGI-like mechanism to remove that rough ER membrane from the host ER and wraps around the rough ER on the vacuole, and that is how it's recruiting ribosomes on it.
Yeah, I have a question here.
Yeah.
Maybe I missed it because of
Yeah.
the data. Do you know what is the site of I cannot say.
Isolation, yeah.
Because it's the ligases and some proteins that
Yeah,
it is
yeah.
picking, but not all. So
Yeah.
did you show that and I
No,
missed it?
I didn't show. We can only identify the protein, but we've tried very hard to look specifically at which residue it gets manipulated. The problem we are facing is technical, which is that once you have that second fragmentation, the manipulation is getting off. Like it's not getting stable enough to hold it during the
So in the
second.
mass spec, you're
Yeah.
losing the manos when
Yeah.
you are trying to do it. Why I was asking is because there is lot of you have to say it for me.
Yeah.
Sites are there.
Yeah.
So if there is a pattern behind it
Yes.
and how
Yes.
it decides it
Completely agree.
and
I completely
the site will be, and it's a enzyme which can do many. It's not, let's call it, you can say semi-promiscuous,
Yes,
right?
yeah, yeah.
So
You're absolutely right. And that is what I think is happening, but we have no data to support that. We know the proteins, it's just the site of the protein. So it could be from the structure. Yeah, yeah. Yeah, we are now collaborating with a lab in Georgia who says that they specialize in glycosylation mass spec. So hopefully we will find some. And I think it's very important to know the sites because that will allow us to understand why is the ribosome stalling, why is the ribosome colliding with this glycosylation.
I think
Yeah. Yeah, so I showed that when we did an unbiased pull down, it binds to a lot of tRNA ligases like VARS, CARS, lysine aminoacyl tRNA synthetase. It binds to a lot of splicing factors that bind to tRNA. So yeah, it definitely binds. How it binds, I don't know. Yeah. Yes, because these are, I know that I'm comparing an RNA molecule with a protein, but it is possible that these proteins have a groove in the tRNA that it recognizes and binds, and that groove is present in CDI by the way it looks. So even though it's not an RNA, it can still bind to these factors, even if it's not in the ribosome. So EF1A, for example, is not in the ribosome, but it can bind.
Okay, so I'll go back to Raj's question. So I was also not 100% convinced that you need both. So I have, so I think the mechanism of blocking, side by binding would imply that the binding affinity would be weak, because then you would need to, to get this protein off quickly to go to the anaerobosome, or it could be regulated by this modification, for example, right? And then you showed the experiment where you have this catalytically inactive mutant that doesn't form the disomes, if I remember correctly. So which would kind of argue against the importance of the blockage by the tRNA, wouldn't it? Because if it's also important, then it should.
No, I get you. I get you. The reason I think that the disome experiment is not foolproof is because I said it's a very, very tricky experiment.
I know.
You need a lot of collided ribosomes to get that small peak. So I feel like even if there is a peak of collided ribosome with the catalytic dead mutant, you are most likely not going to see it.
Yes. So my next point was, so could you actually overexpress, ectopathically overexpress this mutant and see if just by making it more, so basically getting to the 1-to-1 ratio, you would then get the same inhibition and same effect in the disomes as with the active protein that.
Idea. I think that's a great idea. We should do that. I think we have.
So you haven't tried?
We haven't done that, right? So I think the reviewers were mostly asking us to show that there is still a defect with the catalytic dead mutant. And And that we were able to show that, yes, even with a catalytic dead mutant, there's 100-fold. It's not as strong as the catalytic dead mutant.
Can I still have 2 more questions, or the time is up?
Yeah.
So then, with ISR, so the ATF4 is the master regulator of ISR, right?
Yeah.
And you were showing the ATF3. So I'm wondering, so ATF4 doesn't get activated? Because you saw a slide where you also had increase in ATF4.
Yes.
So because I would assume that it has the same thing, right? It also goes up and triggers.
Yeah, you are absolutely right. So I'll just clarify this. I'll just clarify this experiment with the ATF3 and ATF4. Oh, 1 second. Yeah, this one, right? So this is done with anisomycin and not CDI, right? So if you stress the cells with anisomycin and you see the cells, you see ribosome collisions, you see that with an ISR active or an ISR null, you see ATF3, but not ATF4. But if you add tapsigargin, actually, you see both ATF3 and ATF4, right? And in this case, when it's ISR mediated, because tapsigargin does not cause ribosome collision, then both are blocked.
So the collisions, they do not signal through ATF4? That's what this implies?
The collision signals through ATF3.
That I didn't remember. Okay.
So ATF4 will work through EIF2 alpha. But
EN1, and so that's basically the mild stress.
Yes, but ATF3 will not work through EIF2 alpha. So they have 2 different pathways.
And the last thing,
Yeah.
so since there's this mimicry, molecular mimicry, so could you think of an inhibitor, some single molecule, some potent, like a small molecule that you could actually use to specifically block EIF1 binding to the CDI, but not to tRNAs and everything else? So as a potential treatment to because if you prevent this toxin to bind, so the cells should be, the organism should be fine. Have you thought of this?
It is, but Legionella will die with a regular antibiotic. So you don't need any other treatment option. That is why I'm using, just for fun,
I was just wondering, the ribosomes which are recruited near the vacuole versus the one in the cytoplasm, do they have different translation efficiency? Because you said they're actively translating ribosomes.
Yeah.
But then if your protein or toxin is secreted, and I would expect the diffusion to happen near the vacuole.
You are absolutely right. Great observation. So we are right now collaborating with a mass spec specialist who can clearly tell us what are the newly synthesized proteins during infection. So we can see the rate of the translation of these proteins, and we want to know what proteins escape. Like we only found one, which is ATF3, but I'm sure there are other proteins that Legionella is using. And so there's a way to do that through click chemistry, where you can actually find which are the newly translated peptides, and then you can do it during infection and get mass spec results of what proteins are being made at what rate.
But that wouldn't tell which is recruited near the vacuole versus which
but because most of the protein synthesis is blocked, so if it's translating, is most likely on the ribosome. But the problem we have of isolating the vacuole is it's very fragile at that stage. So the cells at 8 hours post-infection are very sick. So if you try to manipulate the vacuole or try to purify the vacuole,
can't you do a single molecule imaging to just look at that?
Yeah, that's possible. Yeah.
Thank you.
In one of your slides, you showed that the position of mannocylation is near the A site or the NPET, targeting ribosomal proteins over there. So is it a possibility that CDI binds with these ribosomal proteins during assembly before it is actually incorporated? On any form of, like, say you overexpress L4 and just do a binding assay and to see at least whether it just binds without it being in a ribosomal context?
Yeah, that's a great point. We haven't done that experiment, so I can't tell you whether it would bind, but we have
avoid the need of EF1.
Yeah, that's a good point. We haven't done that experiment.
Any other questions?
Yes.
One more question. Yeah. So about the ribosome, that what he asked. So if the ribosome is getting recruited even in the vacuole, the EF1 which is getting manipulated, that's still in the cytosol.
Yeah.
So then how some of the EFN are getting
Yeah.
escaped and how
So
some of the
You're
proteins are getting
you're
synthesized?
100% right. And I think that it's not that we are claiming that every single molecule of EF1A is manosylated either, right? So we are seeing effect on translation because there is manipulation of EF1A, there's manipulation of the ribosome, there's manipulation of these. So overall, there's so much manipulation that you're seeing a complete block of protein synthesis, but it doesn't mean that all of these individual ones are completely 100%. We have no quantitative data to suggest what fraction of EF1A in the cells are getting manipulated. So that would answer your question as to how that.
I think you need poison only a small
few, yeah, yeah.
Sort of.
Yeah, we don't think it's 100% per se, yeah.
Okay.
Slightly different type of question.
Yeah.
Given the crazy complexity of this thing,
Yeah.
it raises a question of evolution.
Yes.
What point these different capabilities?
I'm so glad you asked the question because I forgot to mention that ATF3 is present in mammals and in humans, but is not present in yeast. Remember, CDI was killing yeast. CDI was killing amoeba. ATF3 is not in amoeba. ATF3 is not even in yeast. So I think when we looked at the sequence of amoeba, we found a transcription factor that seemed to have this beta zip, sort of fold. And it's possible that in amoeba, it was trying to do something else. And that's why you would think like to cause cell death, why this convoluted process of like going to the ribosome and then colliding the ribosome and then activating a stress response that will cause cell death. In amoeba, maybe it was trying to do something else. And in that amoeba transcription factor has a different outcome than the and that's why in eukaryotes or in mammalian cells, it has chance encountered this and found this. But turns out that what it found is something that the host does when there are ribosome collisions. So irrespective of Legionella, if you just add anisomycin, you see the same response. So Legionella found by chance, I think, a response that was causing cell death, but doesn't mean it cannot cause cell death without ATF3 in other systems. Thank you.